Science is steadily moving closer to the once distant goal of growing fully functional human organs in the lab. This is not a futuristic movie, but groundbreaking research that could someday provide life-saving transplants without the need for human donors. This would reduce waiting lists and eliminate the risk of organ rejection due to donor incompatibility. While some lab-grown tissues and simple hollow organs have already been used in limited human cases, the more complex organs like hearts, lungs, and livers are still years away from routine clinical use. This video outlines where this research stands today, who is leading it, and what challenges remain. It will also look at how much it currently costs, and when these lab-grown organs might realistically become available to patients. Just a friendly reminder to hit the like button and subscribe if you enjoy this video. It would help my channel out, and I very much would appreciate it. Thank you. Now let's get started. Organ regeneration and tissue engineering research is now a global effort, with major centers based in the United States, Europe, and Asia. In the US, institutions like the University of California, San Francisco, or UCSF, Cedars-Sinai Medical Center, and the University of Texas Medical Branch are at the forefront of this work. Europe is also highly active, with collaborative programs such as the European Organ on Chip Society, promoting organ simulation and growth using microfluidics and bioengineering. Asian countries, particularly China, Singapore, South Korea, and Japan, have rapidly increased their investment in this field, driven by both government funding and private venture capital. One notable initiative is China's Organ Reconstruction and Manufacturing Program under the Chinese Academy of Sciences, which aims to become a global leader in regenerative medicine. These institutions and initiatives are collaborating on research that blends cell biology, engineering, robotics, and 3D bioprinting. To date, the most successful applications of lab-grown organs have involved simpler hollow structures such as tracheas, windpipes, bladders, and blood vessels. These structures are easier to engineer because they do not require the complex internal architecture and dense blood vessel networks found in solid organs. Several patients have received such implants under early-stage clinical trials or compassionate use exemptions, showing promising but still limited results. In contrast, complex solid organs like the liver, kidneys, heart, and lungs are far more difficult to engineer. Researchers have successfully grown such organs in the lab and transplanted them into animal models. Pigs are commonly used for these experiments. For example, lab-grown lungs have been transplanted into pigs and sustained function for short periods. However, these organs have not yet been approved or tested for human use on a broad scale. Key challenges include integrating the organ into the recipient's blood supply, ensuring it performs correctly over the long term, and avoiding immune system rejection. As of late 2024, researchers at institutions like UCSF and Cedars-Sinai have made major strides in guiding stem cells to form the complex structures required to build functional organs. Advances in understanding how to manipulate stem cells and control their growth are helping scientists replicate organ-specific architecture more accurately than ever before. These techniques include using 3D scaffolds to shape tissue development, applying mechanical and chemical cues to influence cell behavior, and leveraging gene editing to improve tissue compatibility. While these advances do not yet translate to transplant-ready organs for humans, they represent major steps forward in the scientific pipeline. According to estimates from several leading research centers, lab-grown solid organs may become available for human transplants within the next 5 to 10 years. However, this timeline is highly dependent on continued funding, successful outcomes from ongoing animal studies, and the progression of human clinical trials. Regulatory approval is also a significant hurdle, as authorities like the U.S. Food and Drug Administration, or FDA, must evaluate the safety and efficacy of these new medical technologies before they can be widely used. Furthermore, even if initial clinical use begins within that time frame, it will likely be limited to special cases or high-risk patients. Widespread public availability could take longer, possibly extending into the 2030s, as large-scale production, distribution, and post-operative care protocols are established. The costs associated with lab-grown organs are currently very high, as the field is still in its early experimental stages. In limited human trials, the laboratory cost of producing a tissue-engineered trachea has ranged from $12,690 to $27,490 per patient. When factoring in surgery, hospitalization, and follow-up care, the total cost can balloon to between $174,000 and over $550,000 per patient. For comparison, growing a lab-made lung for animal research costs about $12,000, not including the surgical or veterinary care involved. The 3D bioprinters used in these procedures typically cost around $100,000. The bio-inks and growth materials for each print run add approximately $1,000 more. Despite these high figures, researchers expect that costs will fall significantly as technologies improve, procedures are refined, and manufacturing scales up. Despite rapid progress, no complex solid organ has yet been transplanted into a human in a routine, regulatory-approved medical procedure as of 2024. While initial results from animal models and early trials are encouraging, much work remains to prove that lab-grown organs are safe, reliable, and effective over the long term. There are also biological hurdles that researchers must overcome. Ensuring that engineered organs properly integrate into a patient's circulatory system and function like natural organs is a major challenge. The immune system also poses a risk, as rejection of lab-grown tissue, even if derived from the patient's cells, is still possible. Additionally, the regulatory environment is still adapting to these novel technologies, and approval pathways can be lengthy and complex. Lab-grown organ technology is no longer science fiction. It is a fast-evolving field with real-world applications already underway. Simple tissues and organs like bladders and windpipes have been successfully engineered and implanted, while more complex organs like lungs, livers, and kidneys are in advanced stages of animal testing. Leading institutions across the US, Europe, and Asia are making steady progress. There are many experts who predict that human transplantation of lab-grown solid organs could be possible within the next 5 to 10 years. However, challenges related to function, immune response, regulation, and cost must be resolved before such treatments can become mainstream. For now, the promise remains exciting, but the path ahead is still under construction. So, what do you think? Do you approve of what science is exploring? Leave me a comment, and before you go, check out some of my other videos. Don't forget to hit the like button and subscribe. Thanks for watching, and we'll see you at the next one.